everyone, welcome back to my channel. In this video lecture, we will cover a new chapter of class 11, that is cell, the unit of life. So, what is cell? Unicellular organisms have independent existence and capable of performing the essential functions of life. Anton von Leeuwenhoek described living cell and Robert Hooke described first dead cell. Robert Brown discovered nucleus. Cell theory. In 1838, Matthias Sheldon and German botanists concluded that plants are composed of many different cells. In 1839, Theodore Schwann, British zoologist, discovered plasma membrane in animal, showed cell wall in plants, and told that animals and plants are composed of cells. In 1855, Rudolf Virchow explained omnis cellula e cellula. So, an overview of cell. Cell contains dense membrane, bound structure, nucleus contains chromosomes, cytoplasm is the main arena for cellular activities, different chemical reactions in cell make it living, prokaryotic cell lacks membrane, bound cell organelles, ribosomes are non-membrane bound and found in chloroplast, mitochondria and rough ER. ER means endoplasmic reticulum. In animal centrosomes, membrane, non-membrane bound organelles help in cell division. Size of different cell, mycoplasm 0.3 μm and etc. So, first we'll learn about prokaryotic cell. For example, bacteria, blue-green alga, mycoplasm and PPLO. They are smaller and multiply faster than eukaryotes. Types of bacteria are Bacillus cocci, Spirella vibrio. All have cell wall except mycoplasm. They have naked genetic material and have genomic DNA, that is single chromosome or circular DNA, and have plasmid, small circular DNA outside genome, which gives resistance to antibiotics. They help inclusions bodies and also have mesosomes, enfoldings of plasma membrane. Then cell envelope and its modification. It is present in most of the bacterial cells. It includes glycocalyx, cell wall and cell membrane. Cell envelope acts as a protective unit. Glycocalyx could be loose sheath called slime layer or may be tough as capsule. Cell wall prevents from bursting or collapsing. Bacteria can be gram-positive or gram-negative on the basis of their different composition in cell envelope. In some prokaryotes, chromatophores are present which store pigments. Bacterial flagellum is equal to filament plus hook plus basal body. Pili is the elongated tubular structure made up of special protein, and fimbriae are the bristle-like fibers help to get attached to substratum. Mesosomes, it may be form of vesicles, tubules or lamella or cell wall formation. It helps in DNA replication and distribution to daughter cell and etc. Then ribosomes and inclusion bodies. In prokaryotes, ribosomes are associated with plasma membrane. It have two subunits, that is 50 and 30s, that is total 70s ribosomes. Several ribosomes attached to single mRNA to form polyribosomes or polysome. These ribosomes translate mRNA to protein. Then inclusion bodies are several food materials is stored in the form of it in cytoplasm. They are not bound with membrane. Example, phosphate granules and glycogen granules. Gas vacuoles are found in blue-green, purple-green and green photosynthetic bacteria. Then eukaryotic cells, compartmentalization is present. Presence of membrane, bound organelle, cytoskeleton structure. All eukaryotic cells are not identical. Then cell wall. It is a non-living rigid structure, helps in cell-to-cell interaction. Alga cell wall contains cellulose. Cell wall of young plant is primary, which is capable of growth, but secondary wall develops in early as cell matures. Middle lamella is formed of CaZ plus Ca2 plus pectate, which holds cells. Then centriole is present only in animal cell. Then cell membrane. Electron microscope were discovered in 1950s. Structure of RBCs reveal lipids arranged within membrane with polar head outward and hydrophobic tail inner side. Membrane also contain cholesterol. Lipid content mainly consists of phosphoglycerides. Later, it was proved that membrane also contains proteins and carbohydrates. Then, in 1972, Singer and Nicholson gave fluid mosaic model in which lipid have quasi-fluid structure and protein moves laterally within bilayer. This ability to measure as fluidity, which is important for cell growth, intercellular junction, and etc. Now, endomembrane systems, endoplasmic reticulum, Golgi apparatus, lysosomes, and vacuoles. These functions of ER, Golgi apparatus, lysosomes, and gas vacuoles are coordinated, hence they are included in endomembrane system. Endoplasmic reticulum ER divides intercellular space in two compartments, that is luminal inside and extraluminal cytoplasm. Rough ER is the protein synthesis and secretion. They are continuous with membrane of nucleus. Smoother ER, lipid synthesis, steroid hormone synthesis. Then Golgi apparatus discovered by Camillo Golgi. They consist of many flat disc-like sacs or cistern of diameter 0.5 to 1 um, which are stocked parallel to each other. The cysts are concentrically arranged near nucleus with convex cysts forming face and concave trans ways which are interconnected. Performs packaging to deliver outside or inside cells and form glycoproteins and glycolipids. Materials in forms of vesicles are passed to cysts from ER. Processing takes place. Packaged material leaves from transphase. Then lysosomes are membrane-bound vesicular structure formed by Golgi-rich in hydrolytic enzyme. Then vacuoles contain water, sap, excretory product materials which are not useful. And in plant, vacuoles occupy 90% space. They are layered by tonoplast which allow active transport. In amoeba, contractile vacuole perform excretion, and in protist, food vacuoles are formed by engulfing food particles. Then mitochondria, specifically stained, sausage-shaped, cylindrical in diameter. Its inner membrane divides it into inner and outer compartment. Outer membrane forms continuous limiting boundary for organelle, and Christia increases surface area, which is site for aerobic respiration. It is called powerhouse of cell, have associated enzymes. Then matrix is equal to singular circular DNA plus RNA molecules plus 70S ribosomes plus components required for protein synthesis. Plastids is found in all plants and euglenoids, easily available under microscope. Bead pigments. Chloroplasts contain chlorophyll. Chromoplasts contain carotene. Xanthophiles give red, yellow, orange color. Leucoplasts, colorless plastid which store nutrients. Then majority of chloroplasts are found in mesophyll cells. Then grana are also called intergranule thylakoids. Membrane of thylakoids enclose lumens. Then ribosomes in 1953 discovered by George Paladay under electron microscope, composed of RNA and proteins. 80S equals to 60S plus 40S and 70S equals to 50S plus 30S, where S is Swedberg's unit, stand for sedimentation coefficient, that is measure of density and size. Then cytoskeleton, network of filamentous proteinaceous structure present in cytoplasm involved in mechanism support maintenance of shape of cell. Then microbodies, they are membrane-bound vesicles containing enzymes. They are found in both plants and animals. Cilia and flagella, hair-like outgrowth, and they both are covered with cell membrane. Their core have many microtubules running parallel to axis. Central tubule are connected by bridge and enclosed in central sheath. And sheath is connected to one of the tubule of each doublet by radial spoke. Total radial spokes are 9. Peripheral doublets are interconnected by linkers. Both emerge from centriole-like structure called basal body. Then centrosome and centrioles. Centrosome is an organelle which contain two cylindrical structure called centrioles surrounded by amorphous pericentriolar materials. They are made up of 9 triplet of peripheral tubular protein which are linked. Its center part is hub which is connected to a triplet by radial spokes. They form base of cilia, flagella and spindle fibers. The nucleus, discovered by Robert Brown in 1831 and material stained by basic dye, was named chromatin. Then nucleolus may be one or more. Nuclear envelope consists of two parallel membrane. Pores are formed by fusion of two membrane. Mature RBC and sieve tube cell lack nucleus. Nucleolus is membraneless and site for RNA synthesis. More nucleolus, hence more protein synthesis. Chromatin equals to DNA plus histone plus non-histone protein plus RNA. A single human cell have 2 m long thread of DNA distributed among his 46 chromosomes. Every chromosome has primary constrictions called centromere and disc-like structure called kinetochores. Sometimes few chromosomes having non-staining secondary constriction at a constant location, which gives the appearance of small fragment called satellite. Then chromosomes are divided into 4: metameric, submetacentric, acrocentric and telocentric. Metameric, centromere is middle, 2 equal arms. Submetacentric, centromere slightly away from mid, short and long arm found. Then acrocentric, centromere present near at the end, and telocentric, centromere at the terminal. So this was for this video. Hope you understood it and hope you liked it. Don't forget to subscribe my channel and please like, share, comment my videos. Once more, thank you and please don't forget to subscribe my channel. Till then, bye bye.